All right, good morning to my friends who are joining us on the recording. It is April 2nd. We are covering the second half of lesson number 10. So lots of topics that we want to talk about today. We're going to start with the graph that is at the beginning of lesson 10, and we're going to use it over and over again in lesson 10. So when we look at this graph, the first thing we want to remind ourselves of is that this graph shows me what's called the membrane potential. And I'm going to ask you the same question I always ask. Membrane potential, this potential word is a fancy word for what? What did we say we could trade out potential with? What's the easy word? Absolutely. Yep. Membrane potential, the easy word for it is my membrane charge. Remember that when we talk about the cells of the body, we said that the cells of the body are polarized, which means that the inside of the cell has a different charge than the outside of the cell. And I told you guys during our office hours on Tuesday that there are some important numbers in this lecture. The first important number for us is -70 on this graph here. -70 is the thing that we talked about way back in Unit 1 called the resting membrane potential. The resting membrane potential. So at the very beginning of this graph, when we're looking at a neuron that's not receiving any messages, its charge is always going to be -70. That's always where a neuron starts. The next part on our graph that we see here highlighted with our little lightning bolt, the title of this area on this part of my graph is called stimulus applied. This stimulus word is just a fancy word for saying that this neuron got a message. Messages come in the form of neurotransmitters. Remember from our discussion on Tuesday, neurotransmitters, that's the kind of messages that neurons to each other. So when I'm looking at my graph here, we start off with a neuron that has no messages. That neuron receives some messages. Those messages we're calling a stimulus here, or it's something that's going to cause a change in my neuron's membrane. Let's see if we can remember some of our terminology from our class on Tuesday. When I got this neurotransmitter message right here, it made my membrane charge get more positive. This positive change that I saw right here is an example of a postsynaptic potential. Remember, we had two kinds. We had excitatory and inhibitory postsynaptic potentials. Instead of writing that whole word, right, we had abbreviations. So I'm looking at my graph here. I got a message. That message caused my membrane charge to get more positive. Would I consider this change right here an EPSP or an IPSP? Which kind of change? Yeah, lots of us are chiming in. This would be an example of an EPSP. So somebody typed in the word for me, which is absolutely correct. We got an excitatory message, which means that the change that it caused in my membrane charge is called an EPSP. It made my membrane charge go up. So this stimulus that I got, this neurotransmitter that was released by one neuron to another, that was an excitatory neurotransmitter. The excitatory neurotransmitter changed the charge on my membrane, on my neuron, to make it more positive. It got positive enough, this particular message was excitatory enough that it got positive enough to reach threshold. And remember, we said last time when we were talking about this graph that threshold is an underlined highlight star. We got to know threshold. Threshold is also something we got to know the numbers on again. So over here on the left side of the graph, here's my next number for us to know on this graph. That's -55. When a neuron reaches threshold, that neuron is going to depolarize its membrane. So I'll make a note for us here. If a neuron's membrane, here let's be specific. If a neuron's membrane reaches threshold, the neuron depolarize. The word depolarize means its membrane charge is going to go all the way positive. A depolarized membrane, or a depolarized neuron, excuse me, is a neuron that gets all the way up here to the top part of my graph that I see on here. So the top part of my graph up here, this neuron is completely depolarized. It's completely the wrong charge. But being completely the wrong charge is a good thing when a neuron wants to send a message. So what we want to know about the very top of this graph, there's two things we want to know up here. The first thing we want to know about the very top of this graph is that up here at the top, this is when my neuron can talk to its neighbor. Only when its charge gets all the way up to positive 30 can a neuron talk to its neighbor. The other thing that we want to know, that I just kind of said in passing, is that a depolarized neuron is all the way up. Its membrane charge is positive 30. So 3 numbers we have to know in our neuron physiology lecture. We got to know where we started at, negative 70. We need to know our threshold, which is the charge that's going to allow me to completely freak out, completely depolarize, so negative 55. And then we need to know positive 30, all the way at the very top of this graph. This is the charge I get to when my neuron is talking to its neighbors. When I get all the way up to positive 30, I talk to my neighbors, but I also have a moment of self-reflection where I realize, oh my gosh, I'm positive 30. I'm supposed to be negative 70. This can't stand. I cannot stay here. So we see this part of my graph where very quickly the membrane charge goes back down. The name of the process where my membrane charge goes back down is called repolarization. Repolarization. I'll add a note because we didn't write it before. Over on the left side of the graph, we're looking at depolarization. So going from being negative down here at resting membrane potential, all the way up here, this is the process of depolarization, getting more positive. Once I'm all the way positive up here at positive 30, then I'm going to go through a process called repolarization, going back to my normal charge. But somebody had mentioned, when we were talking about topics for today, had mentioned there is also something that's called hyperpolarization. So I'm going to add that down here because it's down at the bottom of my graph, hyperpolarization. Hyperpolarization is what you see in this region right here, hyperpolarization. So hyperpolarization is, well, it's going back to those beginning words like we talked about before. Hyper means a lot, or in this case, it actually means too much. When I'm doing this process of repolarization, when I'm trying to get negative again, sometimes I get a little too negative. Actually, every time I get a little too negative. Every time I get a little too negative, I'm in this state down here called hyperpolarization. When I'm in hyperpolarization, I am even more negative than where I started at. Pilar asked what the voltage is down there. What I would typically say on a quiz or an exam for hyperpolarization, we're going to call that negative 75 millivolts, about negative 75 down there. It can vary a little bit. For it being hyperpolarized is that it's just more negative than negative 70. So generally the number that I use for hyperpolarized is about negative 75 down here. When I get down here to negative 75, I'm too negative. I don't like that. I'm going to want to get back to my resting membrane potential. There are 2 things that help me to get back to resting membrane potential, and it's things that we actually talked about way back in Unit 1. Or back in lecture number 4 when we were talking about neurons. So there's, 2 things that help me out. The first thing that's going to help me get back to regular resting membrane potential is something called the sodium-potassium pump. Okay, let's see if we can remember this. Not a trick question, I promise. The sodium-potassium pump moves 2 things. What does the sodium-potassium pump move? It's a protein that moves stuff. What are the 2 things it moves? Yep. Yeah, a bunch of us are typing in our abbreviations, which is way faster, right? So the sodium-potassium pump moves sodium and it moves potassium. So the first way I'm going to get back to normal is I'm going to work on equalizing where my sodium and my potassium is. Hey, remind me, was it sodium or potassium that I have more of inside? Was it sodium or potassium that there should be more inside my cell? Yeah, getting a whole lot of potassiums. Absolutely. I should have a bunch more potassiums inside than outside. I should have a bunch more sodiums outside compared to inside. Based on that, I will use my sodium potassium pump to make sure ions end up where they're supposed to be. So the first thing that's going to help me get back to normal resting membrane potential is a sodium potassium pump. But really the star of the show that's going to get me back to membrane potential, our resting membrane potential, are my leakage channels. Leakage channels. My favorite question. Leakage channels are or are not always open. Leakage channels are or always open. Exactly. Lots of us chiming in. A leakage channel, by definition in its name, leakage channels are always open. So the big star of the show that's helping me from being too negative down here to get back to resting membrane potential, the biggest player in that process are those leakage channels. Those leakage channels help me to restabilize my membrane to get it back to my resting membrane potential. So in terms of the numbers, I guess we have 4 numbers that we want to know. We want to know -70 as our normal resting membrane potential. We want to know -55 as the threshold value. If I hit the threshold value, I am going to depolarize my membrane. We want to know +30 because that's where I end up when my membrane is depolarized. The final number that we want to know would be this -75. -75 being I am hyperpolarized, I'm extra negative. So 4 numbers, make sure you know what's happening at those 4 numbers. The kind of things I would ask you about that is what's the value on threshold? What's the value on when a neuron is depolarized? Or I could say that a neuron is at a membrane charge of +30. What would I call that neuron? Would I call it depolarized, hyperpolarized, at resting membrane potential? So just make sure we know those numbers with their names. Let me scroll up. Yeah, I see your question, Rosa, about neurotransmitters. Let me scroll up and look at my other questions too, because I saw that question before. Got the voltage. So Nicole asked, at -55, it is starting to be depolarized. And with that word depolarized, it really just means that the membrane charge is getting more positive. So we're depolarizing even in this place right here, where we're receiving our excitatory message, where we're just going up just a little bit, even before we hit threshold. Any kind of charges that make the membrane charge go up, make it more positive, are technically depolarizing changes. So from the point that we go from resting membrane potential to anything more positive, we've started depolarizing, although we will mainly call this big steep part on my graph up here the part that's the depolarizing when we're doing it really fast. Let me scroll up just a little bit more. Okay, and then we had a question about our gates. So let's work together on the gates question. All right, so I'm looking at my graph here. Well, here I'll put Nicole's question to the class. She said, what would we call it when a neuron is up here at +30? Did anyone catch the word? I would say that neuron is, did anyone catch that? A neuron whose charge is +30. What did Dr. Aulis say we might call that neuron? Depolarized, exactly. My neuron that's all the way up here, completely depolarized. That's a depolarized neuron. A neuron that's somewhere here in the middle, we would say is depolarizing, but +30 is the depolarized. That's as high as I'm going to get on that. So let me tie in to Tuesday's class, because we did talk a little bit about some of our channels on Tuesday's class. What I'm going to do, I think, is I'm going to switch to a different image So if you want a copy of this, all the notes that we took on here, go ahead and take yourself a snapshot on your phone because it's about to go away. I'm going to go to a different image in our notes to talk about our types of channels here. I'm going to go back to this picture that we talked about a lot on Tuesday. This picture shows me what it looks like on a typical neuron when it's receiving a lot of different messages, and those different messages are being added together to give me my membrane charge changes. So on our previous graph, I'll draw like I drew it before. So on the graph we were just looking at, we had resting membrane potential, and then we went up a little bit, and then we hit threshold, and we went straight up, right? Oh, sorry, angle bad. And then once we were all the way positive, we went a little extra negative. Here's what our graph generally looked like before. Pardon my lean there. The part of the graph that we're looking at right now, that we're focusing on, is this little space right here. On the graph that we were looking at, they just showed us a straight line where we received a message and it went straight up. But in real life, it's usually not just going to be I got one message and I go straight up. Usually what's going to happen in real life is I'm going to get a whole bunch of different messages. I'm going to add all of those messages together, and then I'm going to decide if I get to go through depolarization all the way really fast. So we talked about how there are different kinds of messages that neurons receive. Some of them are EPSPs. Remember the E, like we talked about on the previous image, is excitatory. Some of them are IPSPs, I meaning inhibitory. So a neuron is constantly getting messages that make it excited or make it more positive, and messages that are inhibitory that make it more negative. The goal is at the end of adding together all my excitatory and inhibitory messages, we're going to see whether or not I hit threshold. If I don't hit threshold, my neuron's not going to do anything. It's not going to talk to anybody. But if I hit threshold, it is going to talk. Now, we have in neurons, we have two kinds of channels that we care about in neurons. One kind of channel that we care about in neurons are chemically gated channels. The other kind of channel that we have in neurons that we care about are voltage gated channels. Let's see if we can remember from Tuesday's class. All of these changes that I see right here in my membrane charge, were these changes in my membrane charge, were they due to chemically gated channels or voltage gated channels? Which kind of channels cause these changes right here? Okay, so I got a lot of votes. Most of my votes are voting for chemically gated. That is correct for these particular changes that I see right here. Little ones that are making me either a little bit more positive or a little bit more negative. When we talk about receiving neurotransmitters, remember that we said that neurotransmitters are a type of chemical. All of the changes that you're seeing on your graph right here, all of these changes are because my neuron is receiving different chemicals. It's receiving different neurotransmitters. So right here, I received a neurotransmitter that opened up a channel that let some positives in, and I got positive. Or up here, I received a chemical that opened up a channel that either made me lose positives or made me gain negatives. Either way, it made my membrane charge go down. All of these little changes that I see inside here, all my stuff here with my EPSPs and my IPSPs, all of this is because of the specific kinds of neurotransmitter chemicals that I have. Because we're talking about chemicals, we're talking about chemically gated channels. Anytime we are below threshold, so I'll put that here in bright colors here, the kind of channels that I use below threshold. So any part of my graph from -70, I can go even more negative, right? I can get more negative. Anything that's below this -55, all of those changes are due to chemically gated channels because all of those changes are all about those chemicals. They're all about those neurotransmitters. Now, like Harry mentioned for us, things change when we hit threshold, when we hit negative 55. When a neuron's membrane charge goes from being negative 70 all the way up to negative 55, negative 55 means that the charge on the membrane has changed enough that my neuron realizes, okay, we're actually serious. We probably want to depolarize the whole way. So when I hit negative 55, now my membrane charge has changed a bunch. And remember that when we talked about voltage-gated channels open up with charge changes. That's how voltage-gated channels open, is because of charge. It has to be different enough from normal for these kind of channels to open up. And the charge that I have to get to be different enough from normal is negative 55 millivolts. I have to hit threshold. Once I hit threshold, now I open up voltage-gated channels. And those voltage-gated channels will help me to completely depolarize my membrane. Voltage-gated channels also help me to repolarize my membrane to get it back to normal. Because the problem I have at the very top of this graph is that now my membrane charge is all the way up at positive 30. My voltage is completely wrong. So again, it's going to be voltage-gated channels that open up at the top of my graph. And those voltage-gated channels are going to allow me to repolarize my membrane. But remember down here, when I get to negative, at this point, it's those leakage channels that we talked about. The leakage channels are the ones that are going to help me to get back to normal, back to where I want to be. So if we were going to summarize, and we'll hit this graph in just a little bit here. If we were to summarize all this stuff between resting membrane potential and threshold, all of that is my chemically gated ion channels that are involved here. Once I hit threshold, we've got voltage-gated channels that depolarize me, voltage-gated channels that repolarize me, and then I use my leakage channels to get out of this slump where I'm a little bit too negative. Carrie asked if there are particular voltage-gated channels. Yes, there are. And when we get to our graph that has its labeling, we will definitely talk about those particular ones, because it's not just a free-for-all. There are particular ones, definitely. Does a neuron get hyperpolarized by the potassium channels opening too long? Okay, well, we're all asking about it, so let's just go there. Here. Let me find that graph. Let's see. Let's do them in order. So help me out. What page do you have this picture on? This graph where I'm asking you to label the parts on my graph here, page 14. Okay. Robbie, if you're seeing page 12, probably that you are looking at the not updated version of this notes, which is totally fine. Most of the content is the same. But I'll just mention, if you're seeing page 12, probably you're using the one that you printed from lecture class. That will mostly match, maybe not entirely, but mostly match it for you. Okay, here's what we're going to do. Dr. Allis is going to be mean, and she's going to put numbers. Number 4, 5, and number 6. Okay. I want you, hopefully on this graph, okay, so it's either page 12 or 14, regardless. Right now we're going to label our different parts of this graph here. I want you to take a moment, if you have not already labeled your graph, either on page 12 or 14, whichever one has it. I want you to identify the labels on here, because in a moment I'm going to start asking you about specific ones of these regions, and I'm going to have you tell me the number that correlates with it. So each of these lines is representing one of these parts on the graph. Let me give you one quick minute here to make sure you can label it, in case you haven't labeled it already. All right, we have talked about, let's start with, we talked about resting membrane potential. That's my normal membrane charge on my neuron. Which number on my graph, with the numbers that I put on there, which number on my graph shows as resting membrane potential? Which number on my graph, can we tell? 1 through 6. Yep, there we go. Starting to get some numbers in here. The number on my graph that shows as resting membrane potential is here, region number 6. So resting membrane potential is what I see right here at the beginning of the graph. I don't have a line there, but we could also say that right here is also resting membrane potential when my neuron is at -70 millivolts. Remember, that's one of those numbers we got to know, -70, resting membrane potential. I see it over here, labeled over here. What is the name? I'll change it a little bit here. On region number 4, what's the name of this -55 value? What do I call number 4 on here? Yeah, we're texting it in for me. Number 4 on my graph here, this is the place called threshold. Threshold, remember, is when my charge got different enough that voltage-gated channels will open for me. So threshold, very important, because this is when my voltage-gated channels can open. When they open, we get here into region number 3. And in region number 3, my membrane charge is very quickly getting really positive. Do I call that depolarization, hyperpolarization, or repolarization? You can just give me the first few letters. D, hyper, or re. Which one's happening over here? Yeah, that's right. We're all typing in. Over here on number 3, this is when my neuron is depolarizing. It's going from being negative going to being positive. Excuse me. On this side of the graph, we are doing the opposite. We're going from being really positive back down to being negative. On this side of the graph, right here, this part of the graph, would I say that this is a hyperpolarization or repolarization? Hyper or re on this side? Yep, repolarization over here on this side. Number 2 is repolarization. I get a little too excited though. I really want to get back to negative. Uh-oh, I got a little too negative. What do we call it down here when I'm too negative? Down here at the bottom. When I'm too negative, yeah, that's where that hyper word comes in. Hyper means too much. So too negative all the way down here at the bottom. This means, by process of elimination, because I got this question a couple of times, this means that up here at the very top of my graph, this is when we're going to label this process of neurotransmitter release. Neurotransmitter release, the picture, remember that I showed you at the beginning where we had a little text message bubble there? The place in this cycle of the choose my neuron does with its membrane charge, the time when it's going to send a message or spit out a neurotransmitter is up here at the very top. So there are 2 things we could label this one. On the graph, the way I ask you to label it this way, we called this neurotransmitter release, But the other thing I told you that we say is true about this neuron here, we would say that the neuron is completely, you already told me this word once, when my neuron's positive 30, the neuron is completely depolarized. Exactly. It's completely depolarized. The process of depolarization is happening over here. My neuron is done. We get up to here, we're completely depolarized at positive 30. The way, like I said, though, that we're labeling it on this graph is we're labeling it as this is the place where my neuron spits out neurotransmitters. This is where it's going to talk to its neighbor. Once it talks to its neighbor, it's going to repolarize back down to get back to normal. And then, oops, I accidentally get a little too normal, and I'm a little extra negative down here. So the first thing we need to know, be able to do with labeling on this graph is we need to be able to label these parts of the graph, which we just did together here. The second thing we need to be able to do on this graph is we need to be able to identify which types of channels are activated or involved in each of the places. Nicole's asking about neurotransmitter release at the stimulus applied. Neurotransmitter release is when we're talking about the neuron that we're currently tracking membrane charge on. We're back down here. Well, it's hard for me to get back to the graph. This is where it was labeled stimulus applied, right, on the first graph. We would say that this is not the neurotransmitter being released from the neuron whose membrane we're watching. The neurotransmitters that make these changes happen, these neurotransmitters actually came from a different neuron. So they came from a different neuron who got their membrane charge all the way up to positive 30. In this particular neuron that we're tracking its membrane charge, the one we see on this graph, the only time this particular neuron spits out a neurotransmitter is all the way at the top, up here. So here I'll do our day neuron drawings here, right? Really small space. So these are going to be very simplified. Two neurons in a row. They lost their dendrites this morning. It's too early, right? When we are looking at what we're looking at on the graph right here, this graph is showing me the membrane charge of this one. I would have to have a different graph for this one right here. When we're talking about the neurotransmitters that make this neuron change its membrane charge in the way that the graph is showing us, this one released those neurotransmitters, but this neuron, we would also go through and we could do this, we could look at the same graph on this neuron. So this neuron released its neurotransmitters to the neuron that was listening when this neuron got up here too. So kind of an overarching idea is that the only charge that a neuron can release neurotransmitters at is at positive 30. The only charge that a neuron can receive neurotransmitters at or can process them is when we're down here below threshold. Did that clarify with me, Nicole? What other questions do you have about that, or does that make more sense? Yeah, so releasing neurotransmitters is when we pass a message on, absolutely. With both of these neurons change their membrane charge to send a message, and then once this one sent a message to this one changes its membrane charge to be able to send a message as well. So yes, the neurotransmitter is my message. It's when I pass on that message, absolutely. All right, so first we labeled the stages: depolarization, repolarization, threshold, all that good stuff. Now what we're labeling on our graph here is the types of channels that are involved. And so we've said a lot of these multiple times. We're gonna work together to label them here on our graph. So let me give us abbreviations, because otherwise I have to type way too much and you have to write way too much. So voltage gated calcium, voltage gated potassium. We're just doing some abbreviations here, so nothing crazy. Voltage gated is going to be VG. Chemically gated is going to be CG. So when I start labeling stuff on my graph here, don't freak out with these weird letters. And then I'm just going to label my leakage once I'm I'll type leakage. I'll give you guys the word leakage. So, all right, let's start here at the beginning of my graph. Here I am at resting membrane potential, like we just talked about. When I'm at resting membrane potential, I start to receive messages from my neighbors. My messages come in the form of neurotransmitters. Which of my kind of channels that are listed over here, which kind of channels do neurotransmitters open? What are we seeing over here on my graph? Yes, getting a whole lot of my CGs. Absolutely right. These are my chemically gated channels. Now, there are something that we want to make sure we review, because we talked about this on Tuesday. There are three types of chemically gated channels, right? So we had chemically gated sodium channels. We had chemically gated potassium channels. And we had chemically gated chloride channels. Each of these channels can contribute to what's going on here in this region of my graph. When some of these channels open, they make my membrane charge go up. They help to depolarize. When some of them open, they make my membrane charge go down. They're more what we might call hyperpolarizing. Since I talked a lot about that on Tuesday, I'm not going to take a lot of time today to talk about this, but please make sure you remember what happens in terms of which way these ions move and what they do to membrane charge. But the big idea that I see right here is everything in this region. And again, look back at that graph that we talked about where we go up and then we go down and up and down, because that's more realistic. Everything that is right here below threshold, that's always caused by chemically gated channels, always chemically gated. Once I hit threshold, I open up a new type of channel. When I hit threshold, which specific kind of channel did I open up at threshold? Okay, it is a voltage gated. Yep, it's a voltage gated. Which specific kind of voltage gated? Got a few of us chiming in. We know it's voltage gated. It's a VG. It's a voltage gated. Yep, I'm getting a few more answers in with it. Voltage gated sodium. Voltage gated sodium. When I hit threshold, a voltage gated, or not a whole bunch of voltage gated sodium channels open up. They open up because, remember, voltage gated means charge. My charge is different enough, voltage gated sodium channels open up. Those voltage gated sodium channels first open here, and they stay open through this whole process of going up. So the voltage gated sodium channels are important enough that we get to actually label them twice on our graph here. The voltage gated sodium channels are still open when I'm up here. So they open up first. When I first hit threshold, they're going to stay open the entire time as I'm depolarizing. All of a sudden, I hit positive 30. My membrane charge is all the way at the top. When I'm at positive 30, there are actually two kinds of ion channels that I'm going to open up. First basic question here. We're opening them up because my charge is wrong. Which kind of gated was it again that opened because of charge being wrong? Was it CG or VG that opened when charge is wrong? Yeah, a bunch of us are shooting in here. It's always voltage gated. When it's a charge problem, it's always voltage gated. Okay, so the first kind of voltage gated channels I'm going to open up are the kind of channels that are going to help me to get back to normal. The kind of channels that help me get back to normal are my potassium channels. Voltage gated potassium channels. Those get me back to normal. So the first kind of channels I open up at positive 30 when I'm freaking out because, oh my gosh, I'm all the way positive 30 and I should have been negative 70. I open up voltage gated potassium channels. Those voltage gated potassium channels, their job is to repolarize the membrane. So they are also still active over here when I'm in the process of repolarizing my membrane. So voltage gated potassium channels open up when we're all the way up here at the top at positive 30, and they're going to stay open all the way down through this process of repolarization. So voltage gated potassium channels and voltage gated sodium channels, we labeled those two on the graph twice. We used them more than once. There is another type of channel though that also opens when I'm up here at positive 30. This is another voltage gated one. Who opens up at the top when I'm also at positive 30? Who else opens up here? Yes, exactly. My other one, voltage gated calcium channels open up at the top as well. Calcium channels. Voltage gated calcium channels. They bring in a bunch of calcium into my neuron. The thing with voltage gated calcium channels is I don't bring them in. It doesn't have anything to do with membrane charge. For those of us who've worked on this part of the lesson, can you tell me in a short and sweet way, what is the purpose of calcium coming into a neuron? Can anyone remember what happens when a whole bunch of calcium comes inside that neuron? Yeah, Carrie's mentioning my neurotransmitters. Can we be more specific with the neurotransmitters? How does calcium relate to neurotransmitters? When calcium rushes in, so calcium comes in with these channels, calcium enters. It's not an attachment site per se with the neurotransmitters. I kind of like the way that Pilar talked about it a little bit. It helps it to pass the message. When calcium comes into a neuron, that's what's going to make the neuron spit out its neurotransmitters. Neurotransmitters released. When calcium comes in to my neuron, neurotransmitters go out. And I do have a good picture to kind of show this, so we'll come back to this in a minute. But here's the reason why we've got voltage-gated calcium channels up here at the top. My voltage-gated calcium channels up here at the top open up to allow calcium to come inside, because when calcium comes inside, I spit out neurotransmitters. So with these voltage-gated calcium channels, here's a note or an underline highlight star idea I want you to make for yourself about voltage-gated calcium channels. They have nothing to do with membrane charge. They have nothing to do with membrane charge. The ions that matter for membrane charge are potassium and sodium. The purpose of moving my calcium into the neuron, it has nothing to do with what I'm doing with its charge. It only has to do with the fact that when calcium comes in, my neurotransmitters get spit out. So voltage-gated calcium channels open up here. That's going to allow me to spit out my neurotransmitters. Voltage-gated potassium channels start to open up here at the top at positive 30, and we're going to use them, we're going to keep them open all through this repolarization process to get me back down to normal. And then remember, I get a little too normal, a little extra normal, extra negative down here. The last kind of channels, the kind of channels that are going to help me come back from being extra negative down here, which kind of channels are working down here when I'm hyperpolarized? Which kind of channels gets me back to normal? Yeah, left, right? It's those leakage channels. Leakage channels. Those ones are going to get me back to normal. Hey, this labeling that we did on this graph, this is an underline highlight star graph. We got to know this stuff. Which kind of channels are involved in which places on the graph. So make sure we know at the beginning that it's chemically gated channels, because chemicals are attaching to open up these channels. Then we need to know that once we hit threshold, it's all about voltages. The kind of ion that makes my membrane charge more positive is sodium. So voltage-gated sodium channels help me to get more positive. When I'm all the way positive, my neuron is supposed to talk to its neighbor. So this is when my voltage-gated calcium channels open up. But when I'm all the way positive, I also need to get back negative, and I use potassium to get back negative. So voltage-gated potassium channels get me back negative. And it's those leakage channels that very slowly over time get me back up to where I'm supposed to be. What questions do we have still about this graph? Or give me a thumbs up if we're feeling okay about it now that we have it labeled. So give me a thumbs up, or let me know what questions we still have. Okay, well, I'm getting a lot of thumbs up. If you think of a question that comes up later when you're studying, feel free to email me. I'll mention, by the way, I don't know if I'm sitting on emails from some of you guys. I'm a little bit behind on my emails, so I apologize. I'm going to try to get caught up today. So if you have an email that you're waiting for a reply on, please be patient with me. I'm working through those. Let's talk about, I'm going to bounce to this picture here in your packet. Help me out. What page do we see this picture on? Can we help our friends find it? Okay, I'm seeing someone mentioning it might be page 11 in your packet. This picture, I'm going to zoom in and make it a little bit bigger for us here. This picture shows me what happens at the region of a neuron called the axon terminal, or what was called in lab the synaptic knob. It's the same thing. So this is where one neuron is meeting with or synapsing with another neuron. Notice here in my image, I'll pull out here my marker. Notice they have this one labeled the postsynaptic neuron. So this one up here, even though it's not labeled, we could label this one up here the presynaptic neuron. If you're a little bit fuzzy on this presynaptic, postsynaptic neuron terminology, go back to our recording from Tuesday, because we did talk about that. We're looking at the axon terminal here. So this is the end of the road of this neuron. We're at the very end of its axon. This is where one axon talks to another neuron. So one neuron is talking to another. This space where they talk to each other is called the synaptic cleft. The space between one neuron and another neuron, called the synaptic cleft here. Carrie is asking if the channels we're looking at down here are on the cell body of our neuron. Yes, so it's either the cell body or it might be the dendrites. We mainly are going to consider this being part of the dendrites. So we can't see the whole neuron. We're up so close we can't see the whole neuron. We're going to see the dendrites more specifically. So my presynaptic neuron, let's track what's happening up here. Notice that we have our action potential. Now let's recall this word action potential means a change in membrane charge that moves. So I'm going to summarize that as a charge that moves. Action potential is a charge that moves. So at the beginning of looking at here, my membrane charge change is coming down the axon. It gets to the very end of the line with my neuron here. We get to the end of the line here, my membrane charge has changed. At the end of the line, in the axon terminals, I have voltage-gated calcium channels. We should probably underline, highlight, star that. Voltage-gated calcium channels. The reason we should underline, highlight, star that is because the only place on a neuron where we're going to find voltage-gated calcium channels is the axon terminals. I'll say it again because that's worth writing down. The only place on a neuron that you find voltage-gated calcium channels are the axon terminals, the end of the line. Because think about this, when we were talking about voltage-gated calcium channels, we said their function was to bring in calcium, and the job of calcium is to make us spit out neurotransmitters. The only place a neuron spits out neurotransmitters is at the axon terminal, at the very end of the line here. So the only place on the neuron that I'm going to find voltage-gated calcium channels is at the very end, is at the axon terminal. So I bring my change in membrane charge all the way down the axon. It gets to the axon terminal, and when it's down here at the axon terminal, the membrane charge is completely wrong. That's going to cause me to open up my voltage-gated calcium channels. So I can see those here in the membrane of the axon terminal. They bring in a whole bunch of calcium. And when that calcium comes into the axon terminal, all of these little things that we labeled on our lab model, remember these little things down here were called vesicles, or they're little bubbles that are filled with neurotransmitters. Those neurotransmitters chill out inside this neuron until that calcium comes in. When the calcium comes in, these vesicles freak out a little bit. It's like when you're at a party and then somebody comes that you're like, you are my sworn enemy. How did you get an invite to this party too? Like, peace out, I'm gone. So that's what happens here. You bring in calcium and these vesicles are like, nuh-uh, I'm not staying at this party. They spit their neurotransmitters out into the synapse, or into the synaptic cleft, excuse me, into this space where they can go down and they can attach to those chemically gated channels that we were talking about on our neurons. So we go from changing the membrane charge everywhere on the neuron to changing the charge here in the axon terminal. When it changes in the axon terminal, in comes calcium. When calcium comes inside, neurotransmitters leave. They're gone. We're going to spit them out. When I spit them out, that makes them accessible to my postsynaptic neuron, where they're going to go down and they're going to open up those chemically gated ion channels, the chemically gated ion channels. So the big idea that I want you guys to get from this picture is I want you guys to understand the role of the voltage-gated calcium channels. The reason I included this one was just for these guys here, the voltage-gated calcium channels. I only find them at the axon terminal, and their job is to get in that calcium to make those neurotransmitters kick out. Okay. Those voltage-gated calcium channels. Let's go, let me see if it's my next one here. Yep, let's go to this slide here. I am not going to take a lot of class time here to remind you where each of these structures are on the neuron, because I know you've got a good, it's a 2-minute neuroscience video. So literally 2 minutes of your time, you can watch the structures of a neuron. So please do get the neuron structures labeled. Absolutely want to make sure we know the names of the parts. What I want us to spend our time on right now is I want us to spend our time on figuring out exactly where on my neuron I would find these types of channels. So this is, and I'm going to be honest with you here, this is where the rubber meets the road. Like if we can figure out where these channels live on the neuron, we've already figured out what they do on our action potential graph. If we can figure out where they live on the neuron, we're golden, we're set. So we're going to work together here and we're going to try to label our locations of channels. So to help us with doing that, oh, here, I'm going to be a little tricky because you know Dr. Alla, she's pretty tricky here. Okay, the way I'm going to help you guys do that, you're going to tell me the names of the places on my neuron that I find each of these kinds of channels. When you've told me the names, I'm going to type those names down and I'm going to see if you can translate the names onto the pictures. So here's what I mean by that. The first group of channels that we see up here, because we're sorted here in alphabetical order, the first kind of channels we see are our chemically gated channels. There are 3 kinds of chemically gated channels that we care about here. We care about chemically gated chloride channels, we care about chemically gated sodium channels, and we care about chemically gated channels. When I talk about chemically gated channels, what opens them up is chemicals. So we are looking for the places on my neurons where my neuron would be receiving chemicals. Which part of my neuron receives messages or receives chemicals? Exactly. Yep, I'm getting several votes in. All 3 of these ones right here are found on the dendrites. All 3 of my chemically gated channels, only place you find chemically gated channels on a neuron is on the dendrites. And the reason I only find them on the dendrites is because that's the only place that I have chemicals that are interacting with my neuron. So when in doubt, if you're trying to remember where things are, think about what opens the gate. What opens these gates are chemicals. I only have chemicals at the dendrites. Okay, chemically gated channels, I find those on the dendrites of my neuron. Let's go down here. My next one on the list is the voltage-gated calcium channels. We actually just talked about the only place on a neuron that has gated calcium channels. What's the only place on a neuron with those voltage-gated calcium channels? Exactly. Yep, we're chiming in here. The axon terminals. The axon terminals, or I guess the way that I had you labeling it there was the synaptic end bulb. It's the same thing as an axon terminal. So the axon terminal, also known as the synaptic end bulb, That is the only place on a neuron where I have voltage-gated calcium channels. Because remember, the only job of this kind of channels is to help my neuron to spit out neurotransmitters. The only place it spits out neurotransmitters is at the axon terminals. So that's the only place I'm going to find these guys. Then we get to our voltage-gated potassium channels and our voltage-gated sodium channels. Now, voltage-gated means we open with charge, right? So I'm going to use these types of channels to help me do a couple of things. The first thing I'm going to use them to help me do, which was what we're recognizing here, is to move my charge down the axon. So the first place that I'm going to see my voltage-gated potassium and sodium channels at is going to be the axon. I'm definitely going to see it at the axon for sure. Also mention for me the second place that I'm going to see it. The second place that we see voltage-gated potassium channels and voltage-gated sodium channels is at the place called the Axon Hillock. Axon Hillock. That's going to be the second place that we see these kinds of channels. Now, the Axon Hillock, here's a note that we should make for ourselves. The Axon Hillock is the place on a neuron where the neuron does summation. The axon hillock is the place on the neuron where that neuron does summation. Hey, help me out for my friends who were here on Tuesday or who saw the video. What happens when a neuron does summation? Like, what is summation? What is a neuron doing if it's doing summation? I know I'm making you type a little bit. Help me out here. What happens in summation? I like it. Yep, adding. The simplest words is where I add together things. Yeah, we're adding together the EPSPs, my excitatory messages. We're adding together my IPSPs, those inhibitory messages. Summation is the process that I use to figure out if I'm going to get to threshold or not. The place on my neuron where I do summation is the axon hillock. So I'll help you out with your labeling here. This little triangle space right here, where my long axon that sticks out attaches to my cell body, this triangle space right here is the axon hillock. And this is the place where my neuron mixes together all of its messages, where it does summation. Sometimes I like to think of summation kind of like putting things together in a blender. So here's my little blender. I collect all these different messages out here on the dendrites. They change my membrane charge a little bit in all these different places. Mix it together here in my blender. See what comes out. If it hit threshold, I have voltage-gated sodium channels here that will depolarize this membrane. If this membrane in the axon hillock gets depolarized, that will depolarize its next door neighbor in the axon because there's voltage-gated sodium channels there. And if these voltage-gated sodium channels get depolarized, these ones will get depolarized as well. It goes all the way down the axon. That's the process of an action potential. We talked about the way that action potentials worked when we talked about muscles. So if you're not really familiar with action potentials, I probably don't have enough time today to talk about it. Check back in the recordings from last week because I did mention the day that we talked about action potentials. If you have more questions about it, I can take questions on that tomorrow. But the process of doing an action potential in a neuron and in a muscle cell is exactly the same. I change the charge of one part of my membrane, and that causes the next door neighbor part to change, which causes the next one to change, and it goes all the way down the line. Hey, depolarizing my membrane, making it positive, was that the job of voltage-gated potassium or voltage-gated sodium channels? Which one of these types of channels depolarized? Who depolarized it? Potassium or sodium? Who makes my membrane charge go up? Who does depolarization? Yeah, so I'm starting to get my votes in here. Sodium is the ion that I use to depolarize the membrane. So both of these types of channels, voltage-gated sodium channels and voltage-gated potassium channels, both of them are found on the axon hillock and are found all over the axon. The voltage-gated sodium channels open first to depolarize the membrane. But as soon as I've depolarized the membrane, I've got it all the way up to positive 30, those voltage-gated potassium channels will open up and start spitting out potassium to repolarize the membrane. So they're found in the same places because first a voltage-gated sodium channel opens, then a voltage-gated potassium channel opens. Hey, what I will mention, let me see if I can find my picture there for that one. Somebody had asked me, and I kind of ignored the question before, I apologize. Somebody had asked me about this hyperpolarized state down here. Who's responsible for my starting down so low? The type of ion channel that's responsible for my membrane hyperpolarizing, it is those voltage-gated potassium channels. The voltage-gated potassium channels stay open longer than they probably should. They should close for me. If we actually wanted our membrane charge to just go to resting membrane potential and not get extra negative, they would need to close sooner. So they wait too long to close. I spit out too much potassium, and that is why I end up extra negative. So in response to that question from about an hour ago, I apologize, what does hyperpolarize the membrane is that my voltage-gated potassium channels, they stay open too long. If they're open too long, not only will I get to resting membrane potential, I will get even more negative. I'll go down even farther. So the hyperpolarization state is caused by my voltage-gated potassium channels. All right, give me a thumbs up or give me what questions we have. Mainly, I'm soliciting questions about the locations of the channels or about the graphs. I'm going to look at our list here because we got a couple of other topics to discuss here. Okay, I got one thumbs up. I was like, uh-oh, I'm not even getting any thumbs up. Do I have any dancing? Is there anybody dancing? Probably not. It's Thursday, right? It's late in the week. There we go. Awesome. Okay. What I am two dancing ones. Awesome. There we go. Let me go back to the PowerPoint. Yeah, I drew it. So yesterday I almost forgot to draw a penguin. I drew it after a bunch of you had left already. So I'm going to try to remember to draw my penguin today. I got some questions. Emoji penguin. My penguins are way better though, right? Like the ones that Dr. Allis draws are far superior. So I will draw you guys a penguin later in our time today. I got a couple of questions. Okay, here's a question. So spatial summation and temporal summation. Yeah, so what I'll say about that, Pilar, is reference back to the recording from Tuesday. We did talk about spatial summation versus temporal summation in terms of what kind of messages we receive. The short answer to your question is temporal summation is typically just going to be one kind of message. It will either be EPSPs or IPSPs. That's because in temporal summation, remember that all the messages come from just one neuron. Each neuron can only ever send EPSPs or IPSPs. It can only send one type of message. So if we're talking positives and negatives being added together, that's going to be a spatial summation thing. Yeah, Carrie asked, it can't send mixed messages. That's correct. Because neurons can only release one type of neurotransmitter. So they're going to release an excitatory neurotransmitter or an inhibitory neurotransmitter. So either they're sending, if you remember back to our pictures, here, let me find a picture really fast. Right here. Excuse me. When I look at this picture, some of these neurons are color-coded green. All these ones that are color-coded green, they can only ever send excitatory messages. All these ones here that are color-coded red, they're only ever sending inhibitory messages. So really, when we're talking about messages, it's either are you sending your message that is excitatory every time, or are you not sending your message? Or for these guys shown here in red, are you sending an inhibitory message, or are you not sending any message at all? So neurons are kind of committed, unfortunately, for them, right? Because that would get boring. They're committed to either always be sending an excitatory message or always sending an inhibitory message. It's just a matter of, are you turned on or are you turned off? You always only same kind of message. All right, I got the question from multiple people. We're interested in hearing about threshold. And I apologize, I made this really small. Or not threshold, sorry, the refractory period. So let's talk about the refractory period. What page in your packet do we have that refractory period? Can someone help me out? What page are we talking about refractory period on? It's going to be way toward the end. Page 15. Okay. So you should see this graph, and it's probably honestly bigger on your paper than it is here on my slide. So I apologize. 15, 16. Okay, so somewhere in that ballpark there at the very end. This graph is showing me what we call the refractory period. The easiest way to think about the refractory period is a period of time when a neuron cannot respond to a new message, or at least not easily. I'm going to use that clarifier there. During the refractory period in general, what you want to think about with refractory period is a neuron cannot receive any more messages, or it at least cannot respond to any other messages, not very easily. There are two time frames for the refractory period. One of them is called the absolute refractory period. One of them is called the relative refractory period. So when you are reading about and studying these refractory periods, the way I like to remember it is this absolute refractory period. My neuron absolutely cannot respond to any other messages. There's no way. By the way, what happens when a neuron gets a message is it goes through the graph that we were looking at. So it shoots up high and then it goes back down, right? That's what happens when a neuron responds to a message. So during the phase called the absolute refractory period, I can't depolarize my membrane again. I can't get another message from my neighbors. I absolutely cannot. There's also a time called the relative refractory period. During the relative refractory period, it's not that I can't respond to a message. It's just really hard for me to do that. So let's look at our graph here and see these two time periods, and then we'll talk about why we have these two different time periods here. So the absolute refractory period of a neuron, this period happens first. So the absolute refractory period is a period of time from when a neuron first got a message all the way up till about when it gets close to threshold-ish, again, when it's repolarizing its membrane. So the absolute refractory period, it starts when message is received, and it ends around threshold, when I get back down to threshold. So it ends most of the way through repolarization. When I say that a neuron can't respond to a new message, that's kind of my emphasis here. From the point when a neuron first gets a message, let's say I'm talking to you. I send you a message. From the moment that you get my message, the stimulus, to the moment that you send that message to your GroupMe chat members, to the moment when you're getting ready to put your phone back down, let's say. This is what we'd call the absolute refractory period. It doesn't matter if I try to give you another message, you're responding to or working on the first message I gave you. So the absolute refractory period, if I've already given you a message and you're going through the process of sharing that message with a neighbor, if I don't wait long enough and try to talk to you again, you can't hear me. The message came too quickly. The relative refractory period is the period of time where you're still kind of recovering from the first message that I gave you. So the relative refractory period is going to start around threshold. So again, this is when I hit threshold on my way back down when I was repolarizing. It's going to start around that phase, and it's going to end once I'm back to, here I'm going to abbreviate on you, resting membrane potential. The relative refractory period ends when I get back to resting membrane potential. Now, the thing with the relative refractory period is it is possible for me to possibly respond to a new message, but that new message has to be really loud or it has to be really strong. So let's talk about why I have periods where it's really hard for a neuron to get another message. Let's see, for my friends who have done this part of the lesson, can you remember which specific kind of the ion channels that we've been talking about today, which type of ion channel is most responsible when we're talking about the absolute refractory period, when we're talking about not being able to respond to any other messages? Did anyone catch that? Yeah, okay, so a few of us caught that for us. The absolute refractory, I'm not going to type that all. The absolute refractory period is caused by my voltage-gated sodium channels. My voltage-gated sodium channels. Let's see if we can remember this. I'm gonna keep asking it to try to get us to remember this. What did we say the function of the voltage-gated sodium channels was? Did they do depolarization, repolarization, or hyperpolarization? What do these guys do, the sodium ones? Exactly. Voltage-gated sodium channels are the depolarizers. Okay, so here's the problem with the absolute refractory period. During the absolute refractory period, if I wanted to try to respond to a new message, the problem is the kind of ion channels I use to respond to that message, they're already busy. The line is busy. They're already tied up. Because see, from about this time where I start to do summation, where I'm adding stuff together, to when I open up those voltage-gated sodium channels, they stay open this whole time up here. I need my voltage-gated sodium channels to not already be open if I'm trying to use them to make a new action potential. If they're already open, if we're already in the process of getting ready to send a message, I can't reopen more of them because they're already all open. So this absolute refractory period is a problem with those voltage-gated sodium channels. On this side of my graph, they're already open, so I can't open them up again because they're already open. On this side of my graph, when I'm looking at this part over here, I actually kind of have two problems that are going on over here. The first problem that I have on this side of my graph, it's not that my voltage-gated sodium channels are open anymore because we got all the way up to positive 30 and my neuron realizes, oh, I don't need any more sodium to come inside. At this point on my graph, when we're all the way up here at positive 30, what happens with those voltage-gated sodium channels is they actually get deactivated. Deactivated, I'll show you a picture here in a moment. That basically means that they're the wrong shape. Wrong shape. I have this shape where, because they realized that they were really bad, what they did to the membrane charge was like, oh my gosh, we completely freaked it out. They kind of like fold in on themselves, literally, and block the pathway for sodium. So for quite a while, I've deactivated those voltage-gated sodium channels, which means that I can't generate a new action potential even if I wanted to, because my voltage-gated sodium channels are the wrong shape to do that. So the first part of this graph, what's happening is my voltage-gated sodium channels are already open. When I hit this second part of the absolute refractory period, one of my problems going on over, or the main problem over here in the part that's the absolute refractory period, is that my voltage-gated sodium channels are folded in on themselves. They can't open up correctly because they're the wrong shape. Now remember, when my membrane charge gets back close to about threshold, we're at least well on our way to being negative again, we enter what's called the relative refractory period. And the relative refractory period is when it's possible for me to generate a new message, but it's going to be really hard. The reason that the relative refractory period makes it really hard for me to generate a new message is because of this right here. So underline, highlight, star this. My potassium channels are open. Specifically, it's the voltage-gated potassium channels that are still open. And remind me, let's check from before, voltage-gated potassium channels, they depolarize, hyperpolarize, or repolarize. What do the voltage-gated potassium channels do? What's their job? Yeah, so their main job, the main reason we use them is to repolarize. And then they accidentally are so good at their job that they actually end up hyperpolarizing me as well. So yeah, Kathleen, you're they repolarize and they hyperpolarize because they're too good at their job. So notice right here, we're around threshold. We're on our way back down toward resting membrane potential. At this point, when I get down here, my my voltage-gated sodium channels that were in the wrong shape, that were keeping me in this absolute refractory period, they've gotten back to their normal shape. So it's not a problem with the sodium channels anymore. The problem that I have here in this relative refractory period is that my voltage-gated potassium channels, the ones that I use to repolarize my membrane, in other words, the ones I'm using to drop the membrane charge really fast, these guys are open. So they're open here, they're still open here, they don't close until we get hyperpolarized. The idea with the relative refractory period is it's not that you can't get me to make an action potential and go up really high again. The problem is your neuron is actively working on making its charge more negative. So it's kind of like if you were trying to do a high jump, but you've got a 20-pound backpack on your back. Those weights are pulling you down, which is what happens with the potassium channels. They're spitting out ions to make you more negative. That makes it a lot harder for a neuron to go back and go positive. So during this relative refractory period, we would say that the relative refractory period, that's actually a problem with those voltage-gated potassium channels. The reason the problem with those channels is they're working to make the membrane charge more negative, which means if I've got to get back up to threshold, I'm going to have to work harder to make that happen. What questions do we have? Okay, I got a question coming in. Let me read the question. The question says, if the absolute refractory period starts when the voltage-gated sodium channels open, that means when the chemically gated channels send signals at the beginning before threshold, it's not considered the beginning of the absolute refractory period. Well, so to get the voltage-gated sodium channels to open, I was already using the chemically gated channels, because remember, the chemically gated channels get me to threshold. So you could say that, I mean, technically, maybe yes. The problem is we don't see this kind of shooting up on my graph unless I have a signal that's strong enough to push me over threshold. So if you just sent me a really excitatory message, I'm really not going to have a lot of time where there's just chemically gated channels open and not voltage gated channels. That's why, for the sake of our picture here, there's that it's from the moment of the stimulus. This is a really strong excitatory message here, all the way up through this time period. We're going to assume that we almost instantaneously opened up those voltage gated channels. So yeah, thinking about after summation, or like I said, if I have a message that's excitatory enough by itself, I might change that membrane charge right away. If you give me a really strong excitatory neurotransmitter, that opens up enough of my channels right away, and I'm just going to depolarize right away, get those voltage gated channels open right away. Yeah, so technically when we've been looking at the graph, we've focused a lot on summation in this area. For the sake of the refractory period, just assume that summation happened really fast. That maybe that'll make a little more sense for us. Okay, so we want to make sure that we know that the absolute refractory period is a voltage gated sodium channel problem. We want to make sure that we know that the relative refractory period is a voltage gated potassium channel problem. Make sure that we know the difference between what channels are involved here, and make sure that we know that during the absolute refractory period, no new messages. It's not going to happen. During the relative refractory period, if you can get me excited enough, I can respond to a new message. But it's going to have to be a really strong message, because I am dropping down and you're asking me to go straight back up. So I've got to have a really strong message to get there. Yeah, Carrie, we're getting ready to transition into myelin. I just want to show you guys one more thing about these refractory periods to mention for you. I told you with the refractory period, the absolute refractory period, that it came down to a problem with the shape of the voltage gated sodium channel. We've got this picture in our notes showing us the three states that the voltage gated sodium channel can be in. So up here at the top of my picture, let me get my pointer back. Up here at the top of my picture, what the voltage gated sodium channel looks like when my neuron is at resting membrane potential. Remember, the job of the voltage gated sodium channel was to depolarize the membrane. So when I'm resting, when I haven't received a message, when I'm not making my membrane charge positive, my channel, the space for things to pass through, is closed because it's a gated channel. When I hit threshold, I open up that channel. Now I've got space for sodium to rush inside. On the side of your graph where the membrane charge is rising so rapidly, I've got this space here where sodium passes inside, depolarization. But once I get all the way up, to positive 30, I am all the way depolarized and I'm well into my refractory period. So when I have it labeled the refractory period here, we're talking about that part. Let me bounce back to my graph to show you. We're talking, wrong way. Talking about this part right here. When I'm done being open and I'm still in my refractory period, the problem with this side of my refractory period over here is not that my channel is open any longer. The problem is that in this stage of my refractory period, now I've got this little part on my protein. I kind of like to call it the ball and chain part, or think about it kind of like a stopper in a bathtub. When the membrane charge gets really, really positive, positive 30 to be exact, when we get up there, this little stopper goes into place. And so when I told you that the voltage gated sodium channel was deactivated, that's the word we used on the other slide, deactivated. What I meant by it being deactivated is that this protein right here, this stopper protein, goes in and blocks this channel. That is also not going to allow sodium to get inside. So there's two shapes that the voltage gated sodium channel can be in that makes me not be able to open it up again and make a new action potential. Either it can already be open, or it can have this stopper in place. Either way, I'm not getting any new sodium inside. So either way, I am not depolarize my membrane again. So when you get to this part of your lesson and you're looking at the sodium channel, recognize that it can come in 3 different shapes. Only one of those shapes, only this one right here, is the shape that if I'm in this shape, I can make a new action potential. I can spike my graph up again. If I'm already open, or if I've put that protein in, got my stopper protein in place, either of those cases, I cannot shoot back up my graph. I cannot have another action potential. So that's that voltage-gated sodium channel. Let me switch over, because the last thing we wanted to talk about was myelin and the way that I use myelin to send messages. If you still have questions about our sodium channels and kind of the processes with that, please feel free to pop them up in the chat, and I'll come back to them at the very end if we have some time. What we see in our picture here is we're back to our standard neuron that we're used to looking at. They've labeled some of the parts for us. So we've got the big cell body over here. Attached to the cell body is that axon terminal, where I do the process of summation. And then extending off of that is my axon. You know from your studying for lab last week that the axons of a neuron can be covered in myelin. So myelin is a fatty substance. The goal of myelin is to help messages to travel faster. We mentioned in the lab activity that neurons are some of the biggest cells in your body. So your longest cells in the body are the neurons that start in your spinal cord and go all the way to your toes. We have neurons that are that long. So a single cell that is that long needs to send messages quickly. We got to do it fast. The way that we can do that is by wrapping up our axon with myelin. So putting this protective covering around the outside myelin, that's going to help it to send messages faster. So the way that it helps it to send messages faster is by creating just a few places where I have to make my graph spike all the way up, as opposed to making my graph have to spike all the way up across the entire membrane. So let me explain. Let's pull up our picture. And again, I'm sorry I made them so tiny on here. I can go through and edit this. Help me out. What page are we on? This is probably like the last page of your packet here. Yeah, page 16, 17. Yep, that's where the myelin stuff is. So pages 16, 17. Okay. This shows me kind of the 2 options for how the process of sending a message down an axon could work. Remember that a message or an action potential is that membrane charge that moves. That moving charge can move in 2 different ways. One of the ways it can move is something called continuous conduction, and one of the ways that it can move is called saltatory conduction. Saltatory conduction is the way that a neuron that has myelin on its axon can send messages. Continuous conduction is the way that any neurons that happen to not have myelin, this is how they send their messages. So the thing with continuous conduction, and Carrie asked about the door analogy. So when you go to the very end of your lesson and you're studying continuous conduction, it talks about this idea that you are running down a hallway. And when I was writing that lesson, I was thinking about, we're running down the hallways in our empty class building, right? So we're running down Science West. There are 2 options. There is the option that I have to open and close every single door along the way when I'm running down that hallway to help me get down the hallway. So when I open the door, sodium comes inside, and then potassium rushes back into that room, and then I can close it. And then I run to the next door, and I open the next door, and sodium rushes out, and then potassium rushes in. I can close it and go to the next door. If I had to do that with every single door in the hallway in Science West, that would take me a really long time to get down the hallway, because I'm bouncing from the left to the right. I'm going, oh, look, there's 2 doors together. I have to open them up slowly. When we do what's called continuous conduction, there are voltage-gated sodium channels and potassium channels all along the membrane. You see them here on my picture. All of these are little voltage-gated channels. So when I don't have myelin covering my axon, I have voltage-gated ion channels everywhere. There are doors all over the place. Compare that to down here with saltatory conduction. When I do saltatory conduction, I have a myelin sheath. I have a fatty layer wraps around my axon, and that fatty layer does two things for me. The first thing it does for me is it makes sure that all of these spaces here on my axon, it makes sure that there's no ions leaking there. So if you remember those leakage channels that we talked about, the leakage channels are constantly open. They're constantly dripping out sodium and potassium. If I have constant dripping, which is what happens up here, I got some leakage channels that are mixed in between these guys. See how I go from a really strong message, that's the tall part of my message, to a weak message pretty quickly. My message gets weaker faster because I lose some of the differences in my membrane charge. I remake that membrane charge to make sure I don't lose it. So then I go a little bit farther, I have to remake it again. When I look at my myelinated axon, my charge, so my big line here, takes longer to get small, and then I'll regenerate it again in the spaces where I don't have myelin. Here's a lab review for us here. The places where I don't have myelin on an axon are called the nodes of Ranvier. Nodes of Ranvier. That's these little that we see right here. Places on a neuron where I don't have myelin. These are the only places then on this neuron where I might lose ions, where I would have those leakage channels. So I generate a signal, I've got my voltage-gated channels, and then I have this big long space where I'm not losing that signal, it's staying relatively strong until I hit a node of Ranvier. A node of Ranvier is the place where I don't have any myelin. That's also the only place on the membrane where I have those voltage-gated channels. So when my change in membrane charge hits this place right here, now I'll build another charge again. I'll make it really strong, like I see here, and I don't have to do anything with that message again until I hit the next node of Ranvier. So imagine saltatory conduction, or imagine if we're back to Science West, maybe instead of having to go through and open and close every single door in the hallways in Science West, maybe we only have to open and close half of those doors. And we get to go, okay, I opened these two doors, which means now I get to, in the words of the lesson, I get to run 20 feet down the hallway before I have to go in and close another door. So the number of doors or the number of ion channels that I have to open and close, it's a little bit fewer, and they're all concentrated, so I can open and close those doors at the same time, and then I can keep moving on instead of having to go through and do this action potential thing at every single place. I know I had a comment here. Let me go check on that comment. Okay, so Parker said, instead of traveling down the whole axon, myelin allows the message to skip between the nodes of Ranvier. Absolutely. That's a great way to think about it. So sometimes we call saltatory conduction. You'll read about it some places called jumping conduction, where it kind of looks like I jump from one node of Ranvier, because that's where all the sodium rushes in and the potassium rushes out. I don't do anything with that again until the next node of Ranvier. So this whole area here, I get to skip it. I don't have to do anything with that area here. I just make it again here in the next node of Ranvier. So saltatory conduction, we're skipping things or we're jumping between those nodes of Ranvier. So let me go back to my other little neuron picture here. On the axon of this neuron, I would generate the signal here with my voltage-gated sodium channel. I don't have to open up voltage-gated sodium channels again until I hit the right here. I get to skip this whole area here, to use your word. So I get to skip that whole area. Now it's here. And I've got it here. So now I'm going to skip over to this area over here. And now that I'm done here, I get to skip to this one. So it's jumping conduction, or we're skipping parts of the axon. We don't have to cover as much ground. with bringing sodium in and spitting potassium out. Because I don't have to do those things, that's going to allow the message to go more quickly. So yeah, great way to describe it, Parker. That's absolutely correct. Yeah, so Nicole asks, it only stops at the nodes of Ranvier. Yeah, if we're using the analogy of us running down the hallway and opening up doors, yes, the only place in the hallway that you have to stop is at the nodes of Ranvier. We don't have to stop every 2 feet to open every single. There's just one door and then 20 feet and then the next door. So yeah, the idea of stopping at the nodes, like Kathleen mentioned, yes, that's where I would generate the signal. That's where I would generate my action potential or that spike on my chart. So a leapfrog, love it. Yep, we're leapfrogging down the axon. We're jumping down the axon. Instead of having to go every single space, we get to jump some spaces, or we get to skip some spaces. And I know that there is a really cool video, or I think it's cool, that kind of shows the action potential as like a little lightning line going between these little nodes of Ranvier. So definitely check out that little video about saltatory conduction and continuous conduction. I think that'll help with this too. What other questions do we have at this point? Because I will talk a little bit about MS just to give you a heads up. It'll help us understand why myelin's important. Okay, Carrie says she's feeling good. That's great. Yeah, so myelin covers the axon. Myelin is only on the axons, Martha. It's not found on the axon terminals. So once I get to my axon terminals down here, we don't want to have the myelin anymore. We're still going to use all those voltage-gated sodium and potassium channels. So all of my membrane at the axon terminals is going to have to change its charge. There is no myelin down there. Yes, and Carrie, that's right. When saltator is what I use when myelin is on the axon. That is correct. So I'll mention for you, if you happen to have printed the lecture version of neuron physiology, you had a picture that included this half right here, looking at a neuron like this neuron, and then you also had a picture that showed a neuron in multiple sclerosis. Let me see if I can find it online really fast, just to pull up that picture to show you. Let's see. Perfect. Okay, let me see if I can make it big. Maybe not. I'm going to share my screen here and show you guys. I mentioned this earlier too in the class that I pulled this material out of your lecture outline, so you are not responsible for how multiple sclerosis works on the exam or on your homework assignment, but it is a good way for us to think about the role of myelin. So I'll just describe it for you a little bit here in the very end of our class together, just so you can see why myelin's important. So here's the picture that we were using before. We've got our neuron, and it has its myelin sheath that's wrapped around it. I can see it in all of these places here. When I have that myelin sheath, we're doing saltatory conduction, where a signal's made right here, and then it leapfrogs right here, and then it leapfrogs here. So it's jumping between these little nodes of Ranvier. The benefit of jumping is I don't have to signal as many times, but the other benefit of jumping like that, see how this message is kind of being sent here as a straight line. When I only have a few places that I have to remake my message, that message is going to come through crystal clear. There's basically not as much wiggle room. There's a little less margin error there. When we have a neuron that has the process of what's called demyelination occurring, demyelination is when I start to damage my myelin or I even start to remove it. So when you look at the neuron down here, we're looking at a condition called MS, or the actual name for it is multiple sclerosis. So multiple sclerosis is a condition in which the immune system in your body, for whatever reason, thinks that the myelin sheath that we find wrapping around our axon, it thinks that that myelin sheath is an invader. It's a bacteria or it's a virus. Basically, your immune system says, oh, we really probably shouldn't have this in the body anymore. So it starts to go attack that myelin sheath. So notice on here, I've got a couple of places where my myelin sheath is still intact. It's still doing its job. But then I also have several places where that myelin sheath has been really strongly damaged. There are two problems with damaging that myelin sheath. The first problem is one of the goals of the myelin sheath was to block those leakage channels that we talked about. So if I start to damage this myelin sheath, it's possible that some of my ions might leak in and out. That's going to make my messages weaker. So it's going to be harder for those messages to send. But the other thing that might happen is maybe I'm not just going to make my messages weaker. It's also possible that maybe I'm going to start sending the wrong message. So in other words, my neuron gets a little bit confused. Was I supposed to talk to my neighbor? Was I not supposed to talk to my neighbor? I can't really tell because we're up and down all over the place. So we start to get distorted messages or really weak messages that end up traveling down these axons. So in multiple sclerosis, the immune system attacks the nervous system. And to tie this together with our previous unit, let me see if any of these pictures here don't really show it. Well, here, this one might. Let's see. I know it's really small, but check it out. I've got a neuron with damaged myelin sheath right here. And this neuron is synapsing with a muscle fiber. It's synapsing with a muscle cell. See, ultimately, that's the big picture problem with multiple sclerosis, is that we start damaging the myelin sheaths on my neurons that, let's see if this is any bigger. Well, that's smaller. That's worse. Okay. We start damaging myelin sheaths on the neurons that are sending messages to our muscles, telling our muscles to contract. So we start to have issues with really weak muscle contraction or being unable to contract our muscles. So big picture problem with multiple sclerosis. It shows us an idea. Here, I'm going to bounce back to a whiteboard here. Bear with me. Multiple sclerosis. Abbreviated MS. It shows us something that we're not going to have a ton of time to talk about this semester, unfortunately, with losing that week, but it's called the interrelatedness of organ systems. The interrelatedness of organ systems, meaning that if one system malfunctions, we have a lot of other organ systems that need it to be able to function. If one malfunctions, others are going to have problems. So multiple sclerosis, problem number one is that the immune system goes crazy. It's an autoimmune disorder, meaning that you start attacking your own body. Specifically, the part of your body that you're attacking is the nervous system. It's damaged, specifically the myelin. And when the myelin gets damaged, the muscular system can't function right. So we can't contract, so muscle weakness. Big picture, that's what multiple sclerosis, what it was like. So it shows us the importance of myelin. I've got to have myelin on those axons to help them send messages quickly, to help them send messages accurately. If the immune system takes away that myelin, I can no longer do saltatory conduction, and not only do my messages go slower, they might also go wrong. And that's problematic when we're talking about going to a muscle cell. I think we were helping each other out in, let's see, we were helping each other out in the chat. Let me see what it's like. So Lee said demyelination can kind of be like static. Yeah, I kind of like that idea. Think about if I take sheath, I'm getting static like on the radio, and then it's really hard to hear what I'm supposed to hear. I really like that analogy. Parker asked, would we ask a test question about what having more myelin would do for you? I've always heard that Einstein's brain had tons of myelin. So what I will say is I'm not sure if his brain, and maybe his brain had a ton of myelin. This is something that's going to come up here in lesson number 11 for you. We're talking about the brain. What I do know about Einstein's brain is that he had more gyri. Does anyone remember from lab what that word gyrus meant? What's a gyrus on the brain? Yeah, gyrus is a bump. Yeah. So the arrangement of bumps on the brain is not the same for everybody. The number of bumps you have, how big they are, that kind of stuff is not the same. I have heard that he had more bumps in his brain, which would mean he'd have more space for neurons to live. It'd be possible too that maybe he had more myelin as well. So yeah, I like the idea that you asked there about Einstein. I probably wouldn't go that far on the exam. I'm mean, right? But I'm not going to be that mean. So yeah, that's a really interesting thought. What it would be like if we had more myelin, we could definitely send messages more quickly. Absolutely. Yeah, Martha, you asked about the numbers on resting membrane potential. Yes, the one thing I don't like about that last video that I posted for you guys is I don't know where he came up with those numbers. So we are always assuming resting membrane potential is -70. I will not put a distractor on the homework assignments or the exams that's -65 because it's in that video. So -70 is what we're going to assume for resting membrane potential. That's going to be ours. Nicole asked how the myelin sheath got damaged in a multiple sclerosis. Basically, the immune system cells just go and start attacking it. So when they start attacking it, they start to break down. The myelin sheath is actually made out of a cell that's just wrapping around the axon multiple times. So when I start damaging that cell's membrane, that's how we end up with damage to the myelin. I'm not going to ask you about MS on the exam. No, I just wanted to clarify it because a couple of people had asked about it since it was in the lecture notes packet, the original packet. So yes. Yeah, and then some fun chat stuff about Einstein. Yeah. His brain and you know, the more brains that we look at, the more that we examine them, the cooler stuff we can find. I will say too that as you guys know, I am pregnant, obviously, and very recently, one of my pregnancy apps told me that, hey, your baby's brain is starting to build gyri and sulci. And I was like, hey, I'm the anatomy teacher. I know what that means. So that was pretty exciting. So that folding of the brain is something that happens for a really long time to give us space to have all of our neurons that live there on the outside of those bumps. Let me mention one thing. Well, I've got a bunch of you here from all my different lab classes. I had a discussion yesterday with my lab co-coordinator about the sternocleidomastoid. Give me a quick hand if you were in our session yesterday. How many of us are repeats from yesterday? Okay, few people. Yeah, that hand raising, by the way, for anyone who hasn't found it before, it's down underneath your screen. So yeah, probably about half of us are raising our hand that we were here yesterday. Okay, so I had a chat with her because you were there for 3/4. Hey, that counts. That's perfect. So I had a chat with her about the way about that activity, and the question that I had for her and that we kind of discussed a little bit was, does the sternocleidomastoid rotate the skull laterally or medially? Because a case could be made for both. So let's talk really fast about sternocleidomastoid. The sternocleidomastoid muscle, actually, let me go ahead and pull up the lab packet here just so we can look at that together. Open that up and let's find that guy. Sternocleidomastoid is a muscle that, yes, Carrie, I know you emailed me about it. Yep, that's part of why I talked with her about it. Yeah. Sternocleidomastoid muscle. Here, we'll just go to the labeled one because that'll help us out here. Okay. The sternocleidomastoid muscle is the muscle in your neck. Oh, I guess they labeled it not in English. Here's the sternocleidomastoid. Let's put our label on it. Sternocleidomastoid. That's what we're calling it. Sternocleidomastoid muscle right here in your neck. Fun fact about sternocleidomastoid is sometimes it's called the whiplash muscle. So if you're ever in a car accident or you go to a theme park and you get whiplash where you very suddenly turn your head one way, this is the muscle that makes it really hard for you to move your head after that. So the sternocleidomastoid muscle is a muscle that's named based on its attachment sites. We talked about this yesterday in our lab section. The sterno part and the cleido part of its name tells you the attachment sites that are what we call its origins. Origins of muscle are places where they do not move. So the sternum and the clavicle, which the sternum, remember, is in the center of your chest. The clavicle is your collarbone. That's one attachment site. And then the mastoid process is way up here in the skull, a big, massive projection that hangs down. This is what we call the insertion. The insertion always moves toward the origin, which is down lower. We can't see it down here. Okay, so in our class yesterday, when we were talking about the sternocleidomastoid, we drew our origins and our insertions, and then we practiced contracting one side of our neck. So we took our insertion up here at the top, and we moved that insertion toward our origin right here. When we talk about what the mastoid process is doing, when we do this process of moving the insertion up top down to the origin, the movement that we're doing here is moving toward the midline of the body. The mastoid process goes from being really lateral to being more medial. So what I told you guys during our session yesterday is that the sternocleidomastoid medially rotates the sternum, or excuse me, medially rotates the skull. That's what we said in our class, because the mastoid process moves medially toward the sternum and the clavicle. In looking over the way that our assignment is built and the way that it's keyed, we looked not just at how the mastoid process moved, but how the entire skull moved. The entire skull went from looking forward to looking laterally. So on the homework assignment, the action that they're going to say for the sternocleidomastoid is that the sternocleidomastoid rotates the skull laterally. The entire skull goes from being forward to being lateral. So lateral rotation. The way that we were talking about it, because we were focused on origins and insertions, is we said it was a medial rotation because we rotated toward the middle. So here's what I will say, and I ask that you guys please get this out in your group me for me. When you're answering your question about the action of the sternocleidomastoid, the correct answer per the key, because we're looking at the entire skull and not just that insertion, not just the mastoid process. The correct answer that we're going to use for that assignment and for that question is that it rotates the whole skull laterally. The skull goes from being forward to being sideways. The skull also asks you about your bilateral action. When I take both of these insertions and bring both of them toward the origins, it goes down. So when it goes down like that, that is anterior or forward movement, flexion of the skull. So for the sternocleidomastoid, I apologize. Yesterday I did a bad job of explaining it, and I chatted it out, like I said, with the co-course coordinator. So our answer for what the sternocleidomastoid does when just one of them contracts, just one or your right one, that rotates the skull laterally, does not rotate it medially. So I apologize for that confusion. Martha, I will address your question in just a minute. I'm going to wrap up my recording here. Let me just ask before I do, are there any questions specifically about sternocleidomastoid? Because I think that'll be the last thing I put on our recording. Do me a favor, by the way, if you could hit up your class group me's and let everyone know that our correct answer for what the sternocleidomastoid does is it rotates the skull laterally. Just get that message out to all your friends in the group me. Mary Lou's asking, and we did answer this one correctly yesterday. When we talk about the bilateral action, when both of our sternocleidomastoids contract, is that flexion or extension when both of these guys contracted? Who remembers? from yesterday? Did anyone catch it? Yes, it's flexion. Yeah. So when my head gets closer to my sternum, when the distance or the angle between them decreases, that is flexion. That was our definition of flexion. So the angle goes from being this big to being smaller. That's flexion. Yeah. So the two actions when we're predicting our actions on sternocleidomastoid, the first one is that we're rotating laterally. The second one is that we are flexing, or we're bringing it closer together. All right, well, I'm going to end the recording, and then I'm going to stick around for questions. Martha, I will hit your questions. So let me stop the recording. Goodbye to my recording friends. Hope to see you soon.